issue with testing early on. And even now, testing is still a little bit more limited than we would like. And of course, there's the overriding issue of asymptomatic cases. And there's really no way to measure what percentage of that is occurring. Certainly the data from China hasn't been conclusive. So, and we don't have the testing capacity to really understand that. This is something I think is really interesting because it helps you to understand the impact this is having. This is the CDC's weekly morbidity and mortality reports. And the CDC has traditionally done an excellent job of tracking influenza and pneumonia cases every season. The black line is what happens baseline seasonally. The red line is the epidemics that are usually coinciding with flu epidemics every year. And as you can see, if this is 2018, this is 2019, 2020 is definitely a lot different than previous seasons. So that's what everyone's been afraid of, that this sort of thing will overwhelm our hospital system and we won't be able to control the loss of life. This is some really good data just to give you an idea of what's happening locally. So 5,800 cases, about 16% of those cases end up hospitalized. And of the people that are hospitalized, 27% end up in the ICU. So that's a fair portion of people with pneumonia from COVID who are ending up in the ICU. And as we talked about, we're looking at about a 5% case fatality rate. And I thought this might be interesting to you guys too, because so just to orient you, everything from this light blue line and above is adults 50 years and older. And then the green, this darker blue, and this yellow would be younger and middle-aged adults. And as you can see, the percentage of total cases in young people, adults in the 18 to 29 age group, is actually increasing and maybe even overtaking the cases we were seeing in older adults, whereas in the beginning of the local epidemic, it was flipped. But even though there's more young people who are getting sick now, in terms of the total hospitalizations, you're still seeing that the 50 to 64-year-old, the 65 and older group is where we're still seeing the vast majority of hospitalizations. And so it just kind of brings home that there's definitely a demographic of people in our population who are at highest risk and who may not withstand something as severe as a COVID-19 illness. And then we also know that there is a racial and ethnic sort of bias to this infection, that it is disproportionately affecting minority communities. And this is local Rochester data, which really very well mirrors what we're seeing nationally, where African-American and Latinx populations are being affected with both in infection rates that are 3 times higher than their white counterparts, hospitalizations that are almost 5 times higher, and deaths that are, again, 3 times higher than their white counterparts. So there's an age predisposition, there may be a racial and ethnic predisposition. This may have to do with underlying health. It may have to do with exposures. There's a lot of different factors, but we know who's at risk, and that's gonna factor into how vaccine studies are developed and how a vaccine campaign is eventually rolled out.
Angela?
Yep.
In the future, when showing a graphing like that, we may have some people who have no experience in doing graphs like this, so you may have to really walk us through, or at least highlight the key data.
Okay, sure. Yeah. So, I mean, I think the take home is just to eyeball it, really. And if the blue is sort of white patients or white people here in Rochester, the orange is African-American people here in Rochester, the green is Latino or Hispanic people here in Rochester, and the yellow is Asian, then if you look at an eyeball test, and these are cases, these are hospitalizations, and these are deaths, just looking at the eyeball test, you can see that the rate of cases for the orange and the green is significantly higher than for the blue. And that's sort of how we kind of have a sense of who's being most affected, what communities are being most affected by this virus. So how do we get out of this mess? Well, there's two things that you might have heard of on the news. The first is this concept of herd immunity. Um, in the scientific community, we're very mad about how this is being portrayed in the media and the people that are talking about it, because invariably it's not infectious disease scientists that are talking about this idea of herd immunity. And there's a very good reason for that. And then the second potential solve to this issue is vaccination. So I'll talk to you a little bit about what herd immunity means, just to sort of put it into context, and because I just think it's something that's very much misconstrued, and then there's a lot of misinformation there. So this sort of cartoon kind of describes what that looks like. The people in red here on this side are people who have an infection like COVID-19. The people in purple are the ones that are healthy, but they have no immunity to the infection, meaning that they have never been infected with this virus before, and therefore they have no defense against it if they were to see it. So if one person that's infected comes into contact with a bunch of non-infected people who have no defense, then chances are they're going to infect some of these people in the purple. Now, how many they infect is what we call the reproductive rate. For something like measles, the reproductive rate might be as high as 10 to 12, meaning every single person with measles potentially can infect 10 to 12 people who have no defense or immunity against measles. For flu, it's somewhere between 3 and 4. So everyone with flu can potentially infect 3 people who have no immunity or partial immunity or no defense against the flu. And we think with COVID-19 it's similar, that every person that gets a COVID-19 infection potentially will go on to infect anywhere from 2 to 4 other people. So if we were to let this just happen naturally in the world, in our local communities, we were to let the infected people just go along and infect other people, what would end up happening is we'd have a bunch of infected people here in red who now have had COVID-19, and they now have developed immunity or defense against getting COVID-19 again. That immunity isn't perfect. It doesn't necessarily last, but it would give you some protection, and maybe you wouldn't get as sick the next time you got COVID-19. But if we were to let that happen, if, as you can imagine, here in Rochester, we have a case fatality rate of 5,000 people. If you were to extrapolate 5%, if you were to extrapolate 3% nationwide, even here in the United States, we would have millions of people dying, because we would have millions of people being infected. And at a minimum of the 2 to 3% case fatality rate, we would have millions of people dying. That's not a situation that any scientist is willing to countenance in any medical person, which is why you don't hear us talking about herd immunity, natural herd immunity, as a reasonable strategy to decrease transmission of COVID and to halt the pandemic.
Angela?
Yep.
Why would a country like Sweden buy into that concept?
You know, I think that there's a lot of politicization of all of that. I think that if you ask Swedish infectious disease physicians, you'll hear many of them saying that that's really not a reasonable strategy. But the alternative is to do what we did here in New York, which is to close our society, close down our economy, and allow for the losses that are associated, the financial, economic, social losses that are associated with that. It's true that if you do this, eventually enough people will become infected that there will be enough immunity in the community to decrease transmissions rates. We think that about 70% of a community would need to be infected in order for this to have an impact on the pandemic. 70% is an awful lot. And so I think that the experiment in Sweden was not successful. They did see higher mortality rates than their neighbors. And so I don't think that it's one that, at least in the scientific community, we're willing to accept. The contrast to that is vaccination. So again, you have the same two guys in red who are infected. You have these purple people who have no defense. They've never been infected. But now you have this new color, these people in yellow, who get a vaccine. And the vaccine gives them immunity or defense against COVID-19. So that means these two people in red don't really have a chance to spread the virus or transmit the virus to anyone because a large part of your population already has immunity. And the few people that don't have immunity because they refuse the vaccine or they can get vaccinated for some reason, they're still able to be mostly protected because the virus spreads from person to person. So if this red person comes in contact with this yellow person and can't transmit the virus, and this yellow person and can't transmit the virus, and all these other yellow persons surrounding them, then this purple person gets protected because all these yellow people are sort of standing in the way of the virus being transmitted to them. Is that clear?
You mentioned vaccines or herd immunity, but you haven't said anything about mitigation. We're all wearing these masks. I mean.
Yeah. So mitigation is definitely slowing down transmission. But we're not going to be able to wear masks for the rest of our lives until enough people get infected that 70% of the population has been infected. That could take years if we're able to slow down transmission. So it's not a viable long-term strategy. And even with mitigation, in order for this not to have pandemic potential, we'd still need something around 70% of people to get infected. So we just be all getting infected, but at a much slower rate. And the mitigation wouldn't decrease the fatality that's going to be associated with 70% of people getting infected. Okay, so I'm going to talk a little bit about the vaccines, but in order to understand the vaccines that are being developed, I think we have to understand a little bit about the virus. So the first thing to know is that coronaviruses are not new. They've been circulating in humans. We've known they've been circulating in humans since the 50s and 60s.
Angela, I want to just interrupt again. People aren't, as was the fear, this being a cafe, if people have any questions about any aspect that Angela has touched on, please raise your hand and Josh will put you on the screen to ask Angela a question. If there's any concerns, we have a real physician now instead of just some PhD. So if anybody has questions about anything that you're hearing, please raise your hand and Josh will put you on air and you'll be able to talk directly with an expert. Or send me a chat. So please feel free to ask your questions about anything.
Yep, we are definitely ready to start sending in the questions or have you ask the questions. So just either via chat or via raising hand, we're ready to go.
I got one question saying that everything is so perfectly well presented that there are no questions. So maybe you can be a little more ambiguous.
Okay. All right. Well, there'll be questions, I promise. Okay. All right. So coronaviruses have been circulating in human populations for a long time. We've known about them since the 50s or 60s. And the four that I've circled in black here, every single winter and spring, they circulate in communities. They cause colds mostly. Sometimes they can cause pneumonia. And you may not even know you had one. So in the last 20 years, 3 new ones have emerged, which we've never had infections with before. And because of that, they had pandemic potential. The first was the severe acute respiratory syndrome coronavirus, or SARS coronavirus 1. That emerged in 2002 in China. It circulated there for a couple of years, and then it just disappeared. And we're not entirely sure why. But one of the things that it couldn't do was really move from one person to the other. The second one is the Middle Eastern respiratory syndrome coronavirus, and that emerged in 2012. It's been causing local epidemics, mainly in Saudi Arabia, every year since then. But the epidemics are so small that we haven't really gotten a good understanding of this virus. And so far, it doesn't seem to have pandemic potential. Now, what's different about the 2019 novel coronavirus that causes COVID-19 is that unlike these other two viruses, its ability to transmit from one person to the other is extremely efficient. And that's sort of the perfect storm you need to set up a pandemic. A new virus to which we have no pre-existing immunity or defense that transmits very inefficiently from one person to the other. This is a cartoon of the structure of the virus, and the reason I'm showing you this, I promise not to get technical, I couldn't even if I wanted to, is because there's a part of the virus that's really, really important. And that's these little yellow spikes that are sticking out of the surface. In fact, this is what gives the coronavirus its name, because it looks kind of like, it makes it look kind of like a crown when you look at it under a microscope. And this is called the spike protein. And this spike protein is actually the thing that attaches the coronavirus to a body cell. And so it's absolutely necessary for attachment and for infection. And if you could block this from doing its job, you can prevent from happening. So that's exactly what the strategy is for vaccine development. We thought that this protein here, and this is another cartoon, this spike protein attaches onto body's cells, so cells in your nose, in your lungs, in your whole respiratory tract. And once it attaches, the membrane of the virus and the membrane of the cells fuse together, and then the virus kind of pushes its genes into the cell, and the cell makes more copies of the virus, and then the virus gets out of the cell and goes on to infect more cells. So if you can block this part from happening with like an antibody, then you could actually prevent infection, and you halt the possibility of becoming ill from the SARS coronavirus in its tract.
Angela, there was a question about getting back to vaccination and herd immunity. What would, if half the population of the country got vaccinated, would you thought you suggested maybe 70%, but what if only half of the people got vaccinated? Would that slow the spread of the virus, or could that lead to immunity?
So we don't know exactly what percentage of people need to have immunity in order to slow the transmission to a very, very low level. But we know this based on studies done with influenza vaccines. And we know that because the influenza reproductive rate is 2 to 3 people, that the number of people in a community that need to be vaccinated against influenza or have an immunity against influenza approaches 70% to really stop that 1 to 3 people transmission. And so it's then becomes less than 1 to 1. It's not completely gone, but it's less than 1 to 1, and certainly less than 1 to 3. That makes sense. If only 50% of the people get infected, you'll still see a significant number of transmission, but it's still better than nothing, and it still would slow transmission down and certainly slow down the possibility of lots of deaths. Okay, so going back to vaccines and what they're supposed to do. So a lot of people think that when we give you a vaccine, we're giving you something and that thing stays in your body for months or years, and that's what actually protects you. And that's really not true. What the vaccine does is that it trains your body's immune system to respond to the coronavirus if it were to see it in the future. And it does that by mimicking how the body would respond to a natural infection. And there's 2 responses we want to train the body's immune system to do. The first is to induce what we call B cells, and this is a part of the immune or defense system that produces antibodies. So this would be something that looks like an antibody sticking onto the virus, blocking that S protein so that it can attach to the cells. But this also does what we call neutralizes the virus. That means it sort of takes it out of commission. Not only can it not affect, but it sort of just hangs around there until T cells which is another part of the immune system, come by and sort of remove it from circulation. T cells also destroy any cells that may have escaped antibodies and gotten infected with the virus, so that those virus-infected cells can't go on to lead to infection of other cells. So the goal of vaccine is to do those two things, to help your body make antibodies so it's ready to defend you, and also induce T cells to develop memory so that if it were to see the coronavirus, it's ready to activate to destroy any coronavirus-infected cells.
Angela, there was I think a very important question that may be a little outside the context of the talk, but you talked about the virus getting into the cells, replicating, and then being secreted out of the cell. How does the virus cause the symptoms that people are seeing? Why is it, not just a respiratory disease? Why do we see brain effects and COVID toe and all these things?
So, the virus infecting the cells won't function as well. So that's one way that and what's really triggering a lot of the symptoms we feel is that when you get infected with anything that's foreign, that's not supposed to be there, that triggers this whole cascade of inflammation and immune response, which again is your body's natural defense. But that inflammation and immune, while it's trying to destroy the virus, will also destroy your body's natural cells. So in your lungs, you'll get inflammation, and that makes you feel like you have pneumonia, you have problems breathing, you develop a cough. In your liver, in your kidneys, there could be inflammation on your skin surface, which is why you can get things like COVID toes. And so really, the pathogenesis or the cause of the symptoms we feel has less to do with the fact that you have an invading virus than it is to do with how your body is trying to prevent that invasion. Because inflammation and that defense system is both helpful and in some ways hurtful at the same time. With COVID-19, we're seeing inflammation as a response to COVID-19 infection that we've never really seen before. There's a lot of pathology and a lot of disease that's being caused by this just massive inflammatory process that's being triggered by COVID-19 that we don't usually see with many viruses. And that may have to do with the fact that it's completely novel and your body doesn't even know how to respond. And it's really a very deranged response in some ways.
I had another question, which I'm going to read to you. And maybe you may have to interpret this question in plainer language, but the virus would increase its basic reproductive rate if it were less virulent. So the host could contribute to virus transmission for a longer period, all else being equal. Is there any evidence epidemiologically that an increase in this reproductive rate, the R0 rate, or decrease in virulence is occurring as a function of time in America or in any other population in the world? That's
a really good question. Super technical, but it's so I think what he's asking for everyone else is that sometimes viruses mutate to allow them to be able to infect more people and evade sort of detection, evade clearance by the body's defense system. So it'll mutate itself to make it seem less strange to the body, less I'm trying to find a way to explain virulence.
Less toxic.
Yeah, right. So a virus will mutate itself to make it less of an invasion to the body. So to mask itself, to make it seem more innocuous than it is. And that's what we call reducing its virulence. And by making itself seem more innocuous to the body, then that dampens down the immune response. And that allows it to evade that immune response potentially and allows the reproductive rate to increase. Now, is that naturally occurring in the world? We don't have any evidence of that, but it's very early days yet, so I don't know. I do know that small micromutations are occurring, but not to the rate that it appears to affect the pathogenesis of the virus, its ability to affect, to cause disease, the type of disease it's causing, its overall virulence. I do think that with vaccine, the pressure of vaccine could bring that out. So that's certainly something that we're worried about. It's partially the pressure of vaccine that does lead to flu virus changes every year. So it's certainly possible that we could see that in later years, but at this point, there's no real evidence of that yet.
Does the innate immune system play any role in the body's protection against natural protection against COVID-19 infection, or could the innate immune system be targeted to attack the virus?
You know, I'm sure that the innate immune system does play a role. Certainly, once you're naturally infected, the innate immune system is triggered. To date, we actually are trying to suppress innate response because it does cause a tremendous amount of inflammation. And while that inflammation is there to try to stop virus from replicating itself, it's a very fine line between that period when the virus is replicating and the period when the virus maybe is not replicating that much, but the inflammatory cascade has been turned on and it can't be turned off anymore. It's just so deranged that people get strokes and they develop blood clots and their hearts are affected and their lungs are affected and they're on ventilators. And so at this point, you know, the innate response is a bit of an enemy sometimes.
I see.
So Angela, we have a question from Victor Polishuk.
Yeah.
Yeah, my question is, once you've sensitized the B cells and the T cells to the coronavirus and time passes, is the predictability of an anamnestic response there? I mean, how do you know that you'll get the anamnestic response that you're looking for once the host is infected or at least exposed to the virus the next time?
You know, we don't know for sure, but I can show you, I mean, yeah.
What's anamnestic? Victor, what is anamnestic?
An anamnestic response is an exaggerated response after the sensitization has occurred the first time around.
Yeah.
Oh.
So it's a really good question. You know, you never know how well the response that you have sort of artificially generated by a vaccine will perform against natural exposure to the real virus. But, you know, the phase 1 and phase 2 data we have, we are, for most of these vaccine candidates, priming the body's immune system with one vaccine, meaning we give you a dose of the vaccine, it does induce antibody and T cell response, and then we're boosting with a second dose to help you develop enough memory of that immune response so that when you do encounter the real virus, that memory can be easily triggered. But there's absolutely no way of knowing how well that's going to work until you actually do an efficacy trial and you see, okay, now people are being exposed to the virus, we've given them the vaccine. Is that memory that you've created after you vaccinate them going to go to work and do the job of protecting them against getting infected? All right. So I have a few more slides, and then, you know, as I said, I think this will be where we get a lot of questions. Initially, when all of this started, the vaccine research and development process, which started sometime around January 11, as soon as the sequence of the virus was released from China to the world, it kind of looked a little bit like this. And this is a story I read in The Economist in April, which I loved because to me, and I was on the inside track of this thing, it felt like we were just throwing a lot of darts, lots of different technology, lots of different vaccine platforms to see which one landed. But it's actually coalesced really nicely around 3 different ways of making a COVID-19 vaccine. I wanted to also talk about one of the big elephants in the room when we talk about COVID-19 vaccines, which is how quickly we're doing this. It is true that in general, vaccines take about 10 years to develop. And the reason is because we spend a lot of time in preclinical development. When we test, we take a product, we test it in the lab for a very long time. We test it in a lot of different animal models. And that process usually takes about 8 years. And that's really the part of all of this that's been fast-tracked, if anything's been fast-tracked. And the reason we were able to do that was because vaccines were already in development for the MERS coronavirus in the Middle East and for the SARS coronavirus 1 from 2002, 2003. So we sort of had a really strong baseline on which to move into adapting what we already knew to the SARS coronavirus 2, and then prepare really quickly for the clinical trials. Now there's 3 phases in clinical trials. The first is phase 1. It usually involves giving the vaccine to about 100 people. And that phase, they typically use 2 different types of constructs of making the same vaccine. Sometimes they'll add extra minerals to make the vaccine work a little bit more efficiently. Sometimes they will use different genetic material, like the entire gene or just a portion of the gene. So they usually have 2 or 3 candidates. And in phase 1, you sort of see which one of those candidates produces the best immune response. Then you move into phase 2. And phase 2 is really where you really make sure that the vaccine is safe. And in phase 2, we tend to give it to about 1,000 people. And we look to see what the right dose of the vaccine is that produces the best immune response with the lowest number of side effects. And again, in phase 1 or phase 2, you're really confirming that there are no serious side effects, and you're optimizing the right dose, and you're also confirming that this vaccine can potentially produce an immune response which will defend you.
Angela?
Then, yeah.
Why when you get a vaccine, this may be, for example, it can be an RNA that encodes a single protein or a fragment of the virus. Why does people get different amounts of pain and inflammation at the injection site? What's that all about?
So pain and inflammation at the injection site is actually evidence that the vaccine produced an immune response. Now no 2 persons sort of inflammatory response to disease or to vaccine or to anything is ever the same. And I think some people just have a very strong inflammatory response to foreign substances like infections or vaccines. And they'll get a little bit more reactogenicity. They'll have maybe a little touch of fever, a little bit more injection site pain or redness. And other people, their inflammatory system is much more subdued and they just won't make that sort of responses. But those reactions aren't necessarily a bad thing. They're really evidence that your body did respond to that vaccine and that it has triggered this immune response, which will hopefully lead to protection.
I mean, the greater the response in the first few days, does that indicate that the vaccine will work better for that individual?
No, it indicates that the vaccine has produced an adequate immune response to start. Whether the vaccine works better just has to do with how well that mimicking of that response to generate antibodies and generate a good T cell response will work. And I think, you were talking about the innate response. And so that initial reactogenicity is usually innate mediated, which is really inflammation mediated. And inflammation recruits cells and immune cells, and immune cells start to trigger more immune cells to be recruited, antibodies to be developed, T cells to respond. So then inflammation is certainly essential to that response, but it's not necessarily predictive of a good response, if that makes sense.
Could you just briefly give us the time course? What happens? You get a needle in your arm, that the immunogen goes into the body, and then how many days does it take for your to select for the B cells to produce that antibody, and then when do T cells start to appear? What's the time course? What's the process?
So that'll start to happen within immediately. So you'll start to you'll have antibodies that B cells that are recruited to that area. You'll start to have antibodies that bind to the substances that constitute a vaccine. But those antibodies initially aren't necessarily selective for that virus or for that protein or for whatever it is you just injected the person with. It's really just the initial response. Over the next 7 to 14 days, then the antibodies become more specific. You develop what we call IgG, and then that also leads to selection for T cells and T cells developing different epitopes, different spots that will recognize parts of the virus and so forth. And so the timeline for really being able to measure whether or not you had the appropriate response to vaccine usually is between 14 and 28 days after the dose. But there is a response that's starting within the first 2 to 7 days. It's just not the specific response to the virus that you're actually hoping to see. You won't be able to tell what that happened until usually day 14 to 28.
Hi, Angela. I had a question of my own, actually. During the different trial phases, are there fixed standards for how safe is safe enough along each step of the process, or is it balanced against efficacy and other factors too?
No. In phase 1 and phase 2, safety is paramount. And so any serious event that is related to vaccine is almost always enough to halt vaccine production, because the numbers are so small, 100 people or so in phase 1, 1,000 people in phase 2. If you were to see a serious event, a serious neurological event, kidney damage, liver damage, heart conditions, anything that would be considered serious that you could then relate to vaccine, that's usually enough to stop a vaccine from being developed, because even if it's 1 in 1,000, that's too much. And some of these trials, sometimes phase 1, almost always phase 2 is placebo controlled. So you're looking to see if there are more adverse events or side effects in the vaccine arm compared to the placebo arm. And if that were true, even if they were just moderate or serious, but they got over them, that again might be enough to stop development of that vaccine. And then, so actually the phase 1 and phase 2, to answer your question, Josh, the primary endpoint is safety. Immunogenicity is usually the secondary endpoint, meaning does it produce an immune response? But efficacy doesn't come about until phase 3. And that's when you put the vaccine in tens of thousands of people. And you see if it does protect them against disease. And by that time, you usually expect that a lot of the safety issues will have been worked out in phase 1 and phase 2. It's not all, because 1,000 people is certainly not enough to be 100% sure, but certainly nothing moves into efficacy unless you felt fairly confident that there were no major safety signals.
So I have a good friend who's a scientist who's older and wanted a vaccine. So they enrolled in a vaccine trial, and he and his wife enrolled, and he got no response in his shoulder, and his wife got an inflammation. So he assumed he got the placebo. He didn't get the vaccine.
Yeah.
So he decided to immediately go and have an antibody test.
Oh, really?
To see if he got it, and if he didn't get it, he was going to join another vaccine trial, but it was just sort of not really ethical. But
I mean, I hear that a lot from vaccine subjects, and it's not a terrible litmus test. It's just not perfect.
So how about these antibody, these serological tests, you know,
Well, I will say the antibody tests are not 100% accurate. They are most likely to show false negatives than they are to show false positives. And
So
so if
they'll
you had a
they'll
negative
miss.
antibody test, I wouldn't necessarily know what to do with that.
So epidemiologists are not using those as much to
No.
predict or determine, to extrapolate how many infections there are as they are the
No, I
virus.
mean, we're using it sometimes in like healthcare workers to see what, you know, how many of them may have gotten asymptomatic infections or been infected. But it's really to rule in than it is to rule out. It's not a good tool in general. And there are so many different ways of doing antibody tests, and it's not at all standardized. So no two antibody tests are equal. And so it's really hard to know how to use those. We generally don't use them except in research purposes, certainly not for case identification or anything like that.
Mm.
All right, I wanted to briefly tell you about some of the vaccines that are moving through phase 1, 2, and 3 trials here in the United States. You've probably heard about all of them on the news, and you've probably heard about this really awful name, Operation Warp Speed, which I think has led to a lot of distrust in the community that's been unnecessary, because it makes people think that we're moving through this vaccine development process with no, at such a rapid pace that we're not giving enough care and time and attention to safety, which is not at all the truth. I think any scientist, any physician who's actively involved in these trials will tell you that we're, you know, our patients are the most important thing to us. And certainly we wouldn't give anyone a vaccine that we didn't have confidence was safe based on the information we knew. But it is a pandemic, and you can't move slowly because thousands of people are dying every day. So, and then the last couple of slides is because people always want to know how vaccines are made, and I'm sure this is very important information for this audience too. I wanted to tell you about the 3 types of vaccines that are currently in phase 1, 2, and 3 trials. So all of these vaccines are one of these 3 types of platforms. Moderna and Pfizer are what we call RNA vaccines, and I'll tell you what that means in a minute. The AstraZeneca, Oxford, and Janssen, Johnson & Johnson vaccines are what we call viral vector vaccines. And Novavax and Sanofi were a little behind everybody else, but those are what we call protein-based vaccines.
What's the Russian vaccine?
I don't actually know. I think it's an mRNA vaccine, but don't quote me on that because I'm not paying attention to that. Okay, so RNA vaccines or mRNA vaccines. RNA is the genetic material that encodes the virus. So in the virus, there's genetic material, and that's called RNA. And mRNA means messenger RNA. So it's basically instructions on how to make genes that are associated with the coronavirus. And these types of vaccines are basically vaccines that contain messenger RNA, which are instructions to make the spike protein gene. Only the spike protein gene, no other part of the virus. But the rationale behind that is that if you give the body instructions to make the spike protein gene in the body, then when the spike protein is made, then the body will develop antibodies and immune response against that spike protein, which primes it for when you see a natural infection or a natural coronavirus, so that you have that immune response that's ready to go against spike protein.
And these viruses will induce the expression of this viral protein in every cell in the body, or is it selective for just the respiratory pathway, the epithelial cells that the virus would normally infect? Is there any selectivity to where this foreign protein is expressed. I can imagine that might not, you might not want that expressed in your brain.
Yeah, no, it's not every protein. I believe it is epithelial proteins that are going to specifically
cells.
express. But again, it's just a protein. And it's a protein that your own body is making. So we're just giving you the gene. Your body's making the protein. And so it's something that while it's recognized as it shouldn't belong there, it shouldn't cause any destruction because it's made and produced by your own body.
Mm-hmm. So one more question that somebody in the group has asked. So with this type of, maybe you need to define what an adjuvant is, but would this sort of vaccine, an mRNA vaccine, an RNA vaccine, require an adjuvant?
No, these vaccines are not adjuvanted. They're just mRNA proteins. They're usually being given within a sort of a lipid layer to protect them as they go into the body. But that's pretty much all the vaccines are made of.
Could you explain what an adjuvant is?
Yes, I will, once we get down to the spike protein made in labs. So the second type of vaccine platform, so the first is mRNA, and that's Pfizer and Moderna. The AstraZeneca and the Janssen vaccines, Johnson & Johnson vaccines, are actually delivering spike protein gene, and you'll notice the thread. All of this is about the spike protein or the spike protein gene. So the Johnson & Johnson and the AstraZeneca vaccine deliver the spike protein gene that's encoded within a different virus called an adenovirus that doesn't replicate in the body and can't cause disease. It's just acting as a Trojan horse to deliver that spike protein gene, again, with instructions for the body to make this protein so that, again, you develop antibodies to the protein. And again, that's exactly how that vaccine is made. No other substances. It's just this adenovirus acting as a Trojan horse and delivering the spike protein gene, which is engineered into that adenovirus so that you can make spike protein and develop antibodies to it. And then the last type of technology being used to make the vaccine is the spike protein itself. So what's happening is in the lab, spike protein is being made synthetically in insect cells, actually, which is super interesting. And this is actually technology used to make one of the flu vaccines that we give to people every year. So we have a lot of experience with this, and it's easy to scale up mass production. The Novavax vaccine has the spike protein that, again, has been made, and you just give spike protein with an adjuvant. And an adjuvant is like a molecule that you give with the protein that in itself sort of triggers an immune response so that you sort of augment the immune response that you're making to just the spike protein alone. The other company that's using the same technology is Sanofi, and they're also adding an adjuvant to their spike protein. So again, this is purified spike protein with an adjuvant, and that's what the vaccine is made of.
All right, Angela, we have another question from the audience.
Yep.
Carolyn, go ahead.
Yeah, in your second, where you use the adenovirus, what does the body do with the adenovirus? Has it seen it before? Does it respond to it? Or
so the two adenoviruses that are being used, the AstraZeneca is actually using a chimpanzee adenovirus that doesn't cause infection at all in humans. So the body has most likely never seen the virus. And within 3 to 5 days, the body will clear the virus away, and it'll just get destroyed the way any sort of foreign substance that gets into your body gets destroyed. The adenovirus that Johnson & Johnson is using is adenovirus 26. That is a human adenovirus, but the seroprevalence, meaning the number of people who've ever been exposed to that virus, is very low. And that's why it was chosen by Johnson & Johnson. So it's unlikely that the majority of us have ever seen it before. And similarly, it's a virus, both of these viruses will not replicate in your body. So whatever is injected as a vaccine, that's the total number of viruses that will ever be there, because the virus isn't making more copies of itself. And within a week, your body's natural sort of immune system will clear it away. Okay. All right, I'm on slide 18. I have 22 to go. We're at 8 o'clock. Can I have some instructions as to what we should do?
You can keep going until we usually run until at least 8:15 if it's okay with you.
It's okay with me, as long as everybody, I want to be respectful of everyone else's time. So
we'll be
this
here.
last question that I put up here, I just thought you guys would, I mean, and it really speaks to a question that was asked earlier, which is how do we know if a vaccine has produced a response that is protective? How do we know that a vaccine is a good candidate and should move into phase 3 trials? And that goes back to what we do in phase 1 and phase 2. So just to give you an example of how this works. So here is the AstraZeneca vaccine. Again, it's a replication deficient chimpanzee virus that expresses that S-glycoprotein because the gene coded within that adenovirus. And in phase 1 study, and this was some of the phase 1 data, the first thing we look for is side effects. And so that phase 1 study was done in a little over 100 people. And this is the kind of data that they'll generally generate and publish or share with investigators. They'll ask after the first or second dose, how many people developed induration at the site, itchiness, pain, redness, swelling, tenderness, and warmth. And then they'll grade it. Mild is yellow, moderate is this peach color, and red and dark red are what you don't ever want to see. So just looking at an eyeball test, you can see that most people did have a little bit of pain, a little bit of tenderness, but it tended to be mild, which means it usually went away within 24 hours. The next thing you look at is how many systemic symptoms, meaning symptoms like fevers, chills, fatigue, headache, joint pain, muscle ache, nausea, are you seeing? And same colors, and a first dose and second dose of vaccine here. And again, it meets the eyeball test again. You're only seeing the light yellow, which is mild, and the peach, which is moderate. If we ever see red or dark red, that's usually something that requires intensive review and may even be enough to halt development of that vaccine.
So that's what happened with the AstraZeneca. There was somebody who developed a severe side effect.
Well, not in phase 1, actually. Someone did develop a severe side effect in the Oxford University UK study, and it was a neurological event. It's hard to know if it was related to vaccine because there's a lot of information there that I'm not actually allowed to talk about. Sufficient to say that the UK felt that it was a rare enough event and possibly not related to vaccine, that the UK study has actually restarted, and our FDA is currently reviewing all of that data to make sure that we can proceed here in the United States now.
Okay.
Is that pattern where the second dose brings up less side effects than the first dose? Is that normal or?
Yeah, it actually is. And this is something we are seeing in pretty nearly all the vaccine studies, that the first dose for like Moderna and Pfizer and AstraZeneca has been the third. The first dose is pretty reactogenic. People do get some side effects that are mild, but about 60% of them are feeling something. And then the second dose, it's more like 25 to 30%. And yeah, so it's much fewer. Mm-hmm.
And we had a question that came through chat. The question was actually, I guess, on the slide before. Do the 3 different approaches to a vaccine have different challenges to delivery? Which are for the easiest or most challenging issues for vaccinating the population?
So I think the mRNA vaccine is probably the easiest to produce because you can sequence and make these genes very, very quickly. So this vaccine has the benefit of being able to speedily mass produce it for the population. Having said that, Pfizer and Moderna have both promised 100 million doses by the end of the year. That's not going to be enough, and I'll tell you why.
So Angela, somebody else asked if if you just inject the spike protein, you can very carefully titrate how much spike protein you administer. But maybe with the genetic, when you inject the genes, maybe you have less control over the amount of spike protein.
Yeah, that's definitely true. But on the other hand, when you inject the genes, you allow your body to make this spike protein in its normal conformation. And so you're manipulating it less. Whereas if you are just putting the purified spike protein, you actually have to stabilize that protein in a way that the area that it's in a conformation that antibodies will bind to it in the right way.
Oh.
The Trojan horse approach is actually the hardest to do, and it's going to take the longest. But it's an approach for which we know that both the antibody response and the T cell response is excellent. And so this probably has the best chance of creating memory response that'll last the longest. And then the spike protein response, the spike protein can be manufactured not as quick as mRNA, but quicker than the Trojan horse approach. And the benefit of the spike protein approach is that we have a really good model of this in Sanofi's Flublok vaccine, which is an influenza vaccine that we use every year. So the manufacturing capability is already in place and has already been worked out as to how to do that. All right, one of the last things I wanted to show you guys is this. So we look at reactogenicity, we talked about that, but the other thing we look at to make sure a vaccine is a good candidate to move forward is that immune response. And what I'm showing you here is the antibody response. Remember, the antibodies bind to the spike protein, neutralizes the virus, prevents the virus from attaching and causing infection. So this is, again, the AstraZeneca phase 1 data. Over here is where their antibodies were at day 0 before they got vaccine. And 3 people, for some reason, had antibodies, but we don't know why. But most of them, as you can see, is down here super low. This is day 28, or 28 days after they get their first dose of vaccine. Over here is sort of the ability to neutralize the virus by antibodies. And you can see some of them are making antibodies that have the ability to neutralize the virus. But then we thought, well, some of them are not making a very good antibody level. Some of them are, but some of them are not. So this is what sort of led to the idea that maybe these vaccines need to be boosted. So at day 28, we give them a second dose of vaccine. And now you see everybody sort of moves into this higher range that we want them at. Why is that the range that we want them at? Because if you look over here in green, this is what we call convalescent plasma samples. These are people who got naturally infected with coronavirus and developed their own antibodies. And as you can see, the mean of that is in this green box here. So if with vaccine, we can produce a mean of antibodies that's similar to what happens with natural infection, then you've achieved your goal. And so that's why the majority of these vaccines are employing a prime boost strategy, where you prime with the first dose and then you boost with the second dose so that you get into antibodies into that level that you really want them to be at, where they're potentially protective.
What about
one question somebody always asks us is, do all the other studies look like this? And I'm very pleased to be able to say that all of the other studies that have phase 1 data, it's looking almost exactly like this. And so at this point, we think they're all good vaccines and no one is sort of pulling ahead of the pack as better than the others.
Angela?
Yep.
So certainly with flu, an older people, don't have their immune system is not as responsive. So older people, my understanding is when they get a flu vaccine, they can get like a triple dose or a larger dose of the vaccine. In the case of COVID-19, where those are the people you really have to worry about.
Yep.
What are the concerns, challenges, or what's being done for older people with regard to immune response? So this is everybody who got it, but
Right.
most important to pull out the older people like myself.
Yeah. So this, so I'm just showing you the phase 1 data, and the phase 1 data for AstraZeneca was just in young adults. But the phase 2 data, I think about a third of them were in adults 60 years and older. And the antibody response is actually really great in older adults. And all of the trials that are being conducted, at least 25% of the participants have to be older adults, because you're absolutely right. We can't have a vaccine that doesn't protect the people who are most at risk. And so far, it's looking promising. But again, it's hard to say because a lot of the difficulties we have in generating protective vaccine responses in older adults may not necessarily have to do with antibody. It may have to do with T cells. And that's something that we're not really going to be able to measure very well until we have an actual vaccine that we're vaccinating people with.
So,
but it's an area of ongoing research and concern.
So we had a question from the group. People may know that when polio vaccine was first administered, there was some actual active polio viruses in there that actually caused people to contract polio. And we've also heard that, there's a lot of anti-vaxxers out there that are concerned about vaccines. How can consumers confirm that commercially mass-produced vaccines are free of these rumored unwanted extras or they're unsafe, or can you address these fears?
Yeah, I think that there are always going to be preservatives and things in vaccines. And so, I don't think that there's data in the literature that supports that preservatives are potentially, the preservatives used for vaccines have led to any of these sort of severe reports of things that have occurred with vaccines. And I think the way the vaccines are being manufactured, it's really very simple technology. It's not meant to require a lot of adjuvants or preservatives or other things. Like purified spike protein is just that, purified spike protein. And adenovirus delivering spike protein, again, is just that. And so, I think we're going to have a hard time convincing anti-vaxxers that these are more safe or equally as safe as the other vaccines. And even if we could, what does that mean to them? But I think for the general public, for the informed public, understanding how the vaccines are made, and I think will go a long way to alleviating fears, which is, why I wanted you guys to see that. So what is the technology being used to develop these vaccines? What is contained in them? Can you get sick from them? Can you get COVID-19 from them? Are they likely to cause neurological disease? Are they likely to lead to other really serious outcomes? I think it's very simple technology. It's smart, it's elegant, but it's pretty simple.
Are we going to have to get these every year like flu virus?
I have absolutely no idea. I will say that, I've talked to a lot of virologists and immunologists that I work with about this. We know that after people are naturally infected, their antibody levels start to wane at about 3 to 6 months. And so it's likely that with vaccine that will happen as well. But it's possible that vaccine may actually do a better job of protecting longer than natural infection will. And so it's really too early to tell.
Why does something like the measles vaccine last for decades, whereas flu changes every year?
Yeah, flu changes every year.
Is that why we have to do it?
The measles, the immunity that measles gives is really complete. With respiratory viruses, like COVID-19 or flu, after you've had an infection, you do have immunity, but it's only partial. and it wanes every year, where it's for whatever reason, it's not the same with measles and some other viruses, particularly DNA viruses. RNA viruses are just a different beast altogether.
When do you think there will be a virus? I mean, I know you don't know, but end of the year, in terms of a vaccine that can be distributed, end of the year, middle of 2021, what's
right. So I think that we will have data that shows one or more vaccine is efficacious, protects against COVID by the end of the year. I know that Pfizer says October. I know everybody has feelings about the election and all the politics involved in it. But I can say that none of these studies have finished enrolling their phase 3 as yet. And so after you enroll, then you have to wait for your outcomes and you have to analyze your data. And I genuinely believe that the end of the year is the earliest we'll have a good sense of that. After we know something works, then we have to do the harder part, which is figuring out how to distribute it, and who gets it first. While the vaccines are being tested, the government has already paid for these pharmaceutical companies to start mass producing it, even if we don't know it works. Because it means that once we know it works or doesn't work, there will be doses available. But there are 330 million people in the United States, and each one of them needs 2 doses of one of these vaccines. And each one of these companies has only promised 100 million doses. And so you do the math, it's clear that if 6 companies give us 600 million doses sometime in the spring or over the next year, it's gonna take a while to get everybody vaccinated. It's gonna take a while before it's widely available. So my prediction is data by the end of the year, some vaccine available in February or March of next year, and widely distributed by the end of next year.
Could you imagine a vaccine that combines both flu and COVID-19 vaccines in the same needle, or would co-administration, or if you get the same vaccine, if you get both these vaccines in the same day, could that cause bad side effects, or could you combine them?
No, I could imagine that. And in fact, we've been working on developing vaccines that could be co-administered. But there's a lot of things that we still need to learn first. Do people really need to be vaccinated every year or multiple times with this? I think everyone will need an additional boost in a year's time. But I don't know if they'll need it seasonally every year for the rest of their lives.
Hey, Angela, I had a question, actually. I was reading somewhere that to the extent that getting the flu vaccine will have an effect on COVID immunity, it's likely to help if it has any effect at all. So we're preaching to the converted with this crowd here, I think.
Yeah.
Is there a chance that getting your flu vaccine will actually help possibly provide protection with COVID?
I really don't see why it would. They're just different viruses, and the immune response that you're developing against the flu, usually against the hemagglutinin protein. I mean, that's just not a protein that's on SARS coronavirus, and vice versa. The spike protein isn't on flu. So I would find that hard to see how that would happen.
So is the benefit,
having said that, definitely get your flu vaccine, because you do not want to get flu this year. Will be the worst year to get flu.
Is the benefit that not getting flu is always kind of a good health outcome? At that
Well,
point?
yes. I think that one of the things we're all really scared of is the possibility of co-infections. Someone who has flu, which in itself could be fatal, and then you layer on top of that the massive uncontrolled inflammation that's associated with COVID. That's just potentially a very, very bad outcome.
Do you have any insight as to the efficacy of this year's flu vaccine yet?
Well, no, because there wasn't very much flu in the Southern Hemisphere. And so because everybody was social distancing so very well. So I think we have no sense yet on how well that works or how well the virus has mutated. If the flu virus has mutated a lot, if there's a mismatch, we don't really know yet.
Okay. So if there's what everybody gets a last shot for questions. Give it just a minute to see if anybody has one last questions. But I think this has just been a expert, beautiful presentation. I've learned a lot. I think I've got some messages indicating that they haven't been asking questions because Angela's doing such a fantastic job. It's great that we have somebody like you at the U of R doing this sort of work. So thank you very much. We really appreciate it. I guess we don't have it's like sports game. It's like sports now, is that we have to pipe in applause, I think.
We do have one more question, I think.
Oh, good.
So Paul Raposa,
Am I unmuted?
you're good.
Okay, great. Angela, a couple observations. In Australia this year, they did report not much flu, but I wonder indeed, with social distancing, if we might not be able to expect the same outcome here. And also if the use of masks maybe cut down the number of viral particles that people are being exposed to, whether it be flu or COVID, so that potentially there may be a slower burn to either or both the diseases.
Yeah, I think you have to take into account the timing of their flu season. The timing of their flu season also coincided with the epidemic in Australia. Mm-hmm. And so Australia was completely shut down during their flu season.
Right.
We're not shut down right now, and flu season is about to get started. And I don't know that we will be shut down when flu hits. So I expect we will have more flu than they do, just because we're not as tightly social distancing as we were, say, in April and May. But it's potentially going to be a tough fall and possibly even a tough early winter. And I think that the local and national administration will respond to that in ways that could both slow flu transmission and COVID transmission this winter. But that's, we have to sort of see how it unfolds. But yeah, I do think our situation right now is really different than where they were at in April and May.
Well, if I could follow that up further, Angela, I think a big part of this is that we have to take into account human behavior and the importance of leadership and helping to generate, particular movements one way or another. I think the last survey that I saw was that almost 50% of Americans would refuse to take a COVID vaccine if it were available this week.
Yeah, I think we're dealing with a lot of misinformation. And, that's why I was happy to speak to you all, because I think hearing from doctors and scientists who are involved in this work will help sort of dispel some of the myths surrounding how fast this is moving, whether the government is moving too fastly and fast tracking things in unsafe ways. I can promise you that on the scientist level, and there are hundreds of us, that's not going to be possible. Somebody will go to CNN if we think that something is being done that's unsafe to our patients and our subjects. And so I think
you go to Fox News.
Right, Fox, CNN, MSNBC. Take your pick.
We've got a red hat in the background there. I don't know what that means.
But I think I think that we can appreciate that there's a lot of misinformation, and that scientists and physicians just need to go out into the community and start talking to people about how we're making these vaccines, how we're ensuring safety, and why you have to get vaccinated, just like we do with flu every year. If we don't do that effectively, people will die, and that's just not an acceptable outcome.
Okay, I think we should close now. It's almost 8:30. I really appreciate you doing this, Angela. Just a fantastic job. I want to just make one announcement, is that we indicated that registration for the cafe would not be necessary, but it turned out it was an administrative requirement this time from the college. We're going to try to deal with that next time. Josh is going to look into that so that registration is not required. People won't have to submit their emails. We apologize that was required this time. We'll try to get a fix on that. And look on the Facebook page, Rochester Science Cafe, or the Rochester Science Cafe blogspot, or via email for the next cafe, which we hope will at least make an effort to do as fine a job as Angela did today. You really did great. I mean, that was just fantastic. And I wish we could give you some applause, but I think everybody was pretty pleased. So thank you very much.
Thank you again for having me.
Keep up the good work.
Stay safe, everyone.
And just to confirm what David said, apologies about registration. That news came down at 4 o'clock this afternoon, that we needed that as part of the process. We'll smooth it out for next month. Thanks again to Angela, and I'm gonna unmute everybody for a little bit of chaos at the end of the meeting. But thank you everyone for coming, and we will be back next month and going on forward with whatever we're calling ourselves now, the Rochester Virtual Science Cafe.
Yeah.
I don't know what it's called, but
I think I want to thank Josh. Josh designed this whole virtual show, and I think it works pretty well. We got a lot of questions out, so thank you, Josh. That was really well done.
And the video will be posted as soon as I figure out how to do that in a convenient way for everybody.
Okay. All right. Thanks, everybody.
Thanks so much, Angela.
Thank you.
Thank you.
Getting applause.
Thank you.
Yeah.
All
Well
right.
done.
Good night, everyone.
It was wonderful.
Good night.
Thank you, everyone. Signing off.
We had a lot of people.
Put the password in the communication.
Yeah, I'm gonna do that next time. I didn't know there would be a password till right before we started, basically. That was news to me.
Yeah.
We'll try to do.
Yeah, I don't know if we can post it online. I think the RIT will forbid me from actually posting the password online, but I'll send it in the email that goes along with the cafe announcement next time.
Yep. Thank you. It's been a real pleasure. I've moved away from Rochester and I've really missed the Science Cafe. So this has been a real treat to see one again.
Yeah, and as long as we keep doing this, then we'll be able to make this available to everybody. So that'll be nice.
Do you have any idea where people are coming in from?
As far as Florida.
Yeah.
Yeah, I'm coming in from Illinois, which is almost as far.
That's not bad. Yes, soon we'll take over the U.S. and eventually the world.
And then the galaxy.
No?
Yeah. Be forewarned that these things take a life of their own. You may need to keep Zooming even when you go back to live.
Yeah.
Right.
Because you'll create a demand.
Right. Right.
I was wondering, well, by the time we go back live, all of the professors who are giving these talks will be experts at giving multimodality lectures to people who are present and people who are remote, and you name it, we'll all know what we're doing by then, or so they tell us.
Yeah, it's a little.
Well, then live lectures.
It's a little bit of a mixed blessing, this, because although it would allow people from many other places to come and attend the cafes, we're a little worried that it may decrease the intimacy of the affair, and that there may be some people, particularly non-specialists, who aren't comfortable asking questions to such a large group of people. So we have to try to find a middle path where everybody can feel comfortable to ask a question regardless of their level of expertise and how well they feel they speak or something like that. So if it gets too big, or that can be a problem. But for, I think, today's presentation, using the audiovisual aids she used, I think we could have had 10,000 people, and I think it would have been excellent.
But you can't give cookies and coffee.
Yeah.
Yeah, I want my cookies.
If it makes people feel a little better, trust me, we're suffering. I'm suffering as much as anybody from the lack of coffee. So we share your pain.
From the lack of coffee.
Yeah. Right. Thank you again, everybody. And we'll post some videos soon and we'll see everybody hopefully.
Oh, I have a question. I couldn't get the slides that she was showing to be very
Oh.
large at all on my screen. I didn't know what to do.
There's different, what device are you using?
I got a Macintosh and I'm using Zoom.
Oh, there's a view button in the upper right, hopefully, with a
Yeah.
walk type thing.
There's a screen with like six, with a grid of a number of dots, and then there's a
Right.
open kind of frame.
I saw that. The individual people, I could make that screen much larger, but I couldn't get her slides bigger.
If you can see the slides as well as other people, there's usually a slider between the two. And if you slide it away from, if you slide it to make the slides bigger and the people smaller, that usually helps maximize things.
Okay, I tried to ask in the beginning, but I couldn't get unmuted.
Gotcha.
Oh,
Also-
and when in doubt, send things in chat because that's the that's
Okay,
easier back channel.
okay.
And under the view option during a presentation, there's a swap menu item that swaps the presentation with the people.
Ah, where is that?
Well, it's not there now because nobody's presenting.
Oh, I see.
we'll try to make a note of that and make that, we'll cover that, we'll workshop that at the beginning of the next cafe.
Okay, thank you.
Yeah, thanks for that comment. We appreciate it. That's important. I'm sure many other people had that issue, you know. What is that, a barometer in the back or something you have there?
A barometer in the back?
Is that a clock over to your left shoulder?
Oh, this?
Yeah.
It's a thermometer and a hygrometer.
Ah, okay. Okay.
It tells me why I'm uncomfortable.
Okay. All right, everybody. Good night. Very good.
Good night, everyone.
Thank you all.